All right, so here we're going to move on and talk about the digestion of carbohydrates. We're going to continue our story. And this time we're going to focus on disaccharidases. The disaccharidases are going to be at the brush border. So these are just a picture of your mucosa and submucosa of your small intestine with its villi. And then your brush border, of course, is going to have many villi. It's where you're going to contain transporters, absorption of nutrients, which includes your sugars. So your disaccharidases are located in the intestinal brush border, and they're considered enzymes of the brush border. They are going to convert your disaccharides to monosaccharides, and they are glycosidases. Glycosidases that we're going to talk about are going to be in 4 different glycoproteins. We have our glucoamylase, our sucrase maltase complex. We have something called our trihalase, and then a lactase glucosyl ceramidase. So this is just a table that talks about the different complexes and what they target. And so we are going to have our beta-glucoamylase, and this is going to work on our polysaccharides. And this time it's going to split our alpha-1,4 glycosidic bonds, and it's going to occur at the non-reducing ends of the chain. Recall that alpha amylase also targeted our glycosidic 1,4 bonds, but only internally. It was an endoglycosidase. And so this is an exoglycosidase. It's at the ends. Now there's going to be an alpha glucosidase, beta glucosidase. They both have similar functions. They just have different targets. We're also going to have a sucrase isomaltase. And sucrase is going to be able to split up sucrose, maltose, and maltotriose. The isomaltase complex is important because it's the one that's going to break down any branches. So if you have the 1,6 bonds, it's going to be able to split those, hydrolyze those, as well as the 1,4 bonds. We have our beta-glucosidase complex, which is going to be able to break down sugar molecules that are going to be bound by lipids or ceramides. And so we can have glycolipids, glycoceramides, etc., and those can be broken apart so you can free up your sugar to be absorbed. In this complex is also lactase, and lactase is going to split that beta-1,4 bond between glucose and galactose. Trihalase is going to split a anomeric bond, so this is an alpha-1,1 link. So that's the link through the anomeric carbons. Okay, so let's just talk a little bit about what this looks like. So remember, we started out at the top with our starch. So we had a starch molecule. And then this was worked on by your alpha amylase. In both the saliva and the pancreas. And then alpha amylase worked on this, and then you were left with glucose molecules that are amylose, and also amylose pectin. With the branches. And then you're going to have alpha-glucoamylase work on those non-reducing alpha-1,4 bonds. Okay, so this is the non-reducing. And remember up here, alpha amylase was going to be on the reducing. And it's an endo, so internal. And then what you're going to be left with is you're going to have our limit dextrins. And you're also going to have your maltotriose and your maltose. And maltose, which is a disaccharide. And all of these are going to now have to be metabolized by other enzymes. And so your limit dextrins, maltotriose and maltose, can all be metabolized by your sucrase isomaltase complex. And they will come out as a glucose. And remember, the sucrase isomaltase complex, the isomaltase portion, can do the alpha 1,6 and it can do alpha 1,4. These will also be able to go through your maltose triose, and maltose can also go through the beta glucoamylase. And again, you're going to come out and you're going to have glucose. Your sucrose portion up here, or sucrase, this is going to be able to take your sucrose, which is glucose and fructose, and you're going to feed it through here, and you're going to get a glucose out, and you're also going to get a fructose. And then on the other end, you're going to have the beta-galactosidase complex. And the beta-galactosidase complex is going to have lactase. And this is going to do the beta 1,4 bonds. And so you're going to have lactose. And I should put here, this is sucrose. And your lactose is, of course, going to be glucose and galactose. And so this is going to feed through your lactase. And you're going to come out with a glucose. And you're going to come out with a galactose. And also in this beta-galactosidase complex, you also have your ability to break down your glycolipids, including glycocerebosides. So again, you have your glucose, and this time it's going to be bound to ceramide, for example. And then this can enter, and it'll come out and give you another glucose. And of course, it will release ceramide. Now this is given a name. And for it to be able to remove your lipids or your ceramides from your sugars, it's a phyllorhizin hydrolase. Now we have done this and we have generated our 3 monosaccharides, our galactose, our fructose, and our glucose. And these are the 3 that are going to get absorbed. But let's just look a little bit more at these complexes. So what to remember is that the complexes are going to have multiple catalytic sites. So they're going to be globular domains with their catalytic sites. And so here we're showing our sucrase isomaltase, and we're going to have the isomaltase site, and this is the one that's going to do the alpha 1,6. So that's the isomaltase activity. And it can also do the 1,4. And then we're going to have the sucrase activity that is going to be able to split up sucrose. Now you also have your maltase activity that can break the 1,4 bonds as well from your isomaltase. So sometimes you'll see this complex as sucrase maltase complex, and sometimes it'll be labeled sucrase isomaltase complex, but they're the same thing. Now we also have our beta glycosidase complex. This is the one that's going to have the ability to split lactose as well as remove ceramides or remove fats from your sugars. And so this is going to have that glucose ceramide complex, that fluorizin hydrolase. This is also the complex that will allow you to hydrolyze artificial sugar substrates. And then finally, we have trihalase. This is only going to have one catalytic site, and it's going to target the alpha, the anomeric carbons, the alpha 1,1 bond. And so this is not an important enzyme in the United States because trihalose is only found in insects, fungi, and algae, and this is not a huge dietary component of us. But it is still an enzyme that humans do have. Now finally, we're going to have your colon bacteria in the colon that are going to be able to pick up your sugars and digest them as well. So we're going to have those starches that we started out with that got broken down, and you're going to have some that are not very well hydrated, such as your dried beans, and these will get passed on to your colon where your bacteria will metabolize. Dietary fiber and sugar that simply were not absorbed or not broken down get passed into the colon. And the colic bacteria are going to form gases, short-chain fatty acids, and lactate. Some of the fatty acids that are going to be formed are acetic acid, propanoic acid, and butyric acid. And these can be absorbed by your mucosal cells in your colon and be used by the colon mucosal cells for energy directly. Some of the gases, not surprising, are going to be hydrogen, carbon dioxide, and methane.